Look at any standard model of Alzheimer's disease, and you will see time, specifically chronological age, treated as a primary risk factor. We model the disease as if the ticking of a clock is itself a biological mechanism. But at the resolution of a single cell, a clock reading cannot cause a disease. The phrase age-dependent is an unpaid promissory note. It is a placeholder for a specific physical cellular lesion that we have failed to name. That missing physical state is cellular senescence. This is the exact definable cell biology that stands in for the vague concept of aging. Senescence is a stable, irreversible exit from normal cellular function. When a cell sustains too much macromolecular damage to continue its regular program, but refuses to undergo programmed cell death, it enters this withdrawn state. If the cell simply sat there quietly, it would be a harmless, isolated retirement. But the senescent cell is highly active. It adopts a profile known as the senescence-associated secretory phenotype, or SASP. The SASP turns a damaged, non-functioning cell into a broadcaster. It actively secretes a dense, toxic cocktail of pro-inflammatory cytokines, chemokines, and matrix-degrading proteases into its immediate neighborhood. This newly formed secretome provides a biological bridge. It allows us to hypothesize how the private, isolated erosion of aging might eventually travel through surrounding tissue. To understand how this collapse travels, we trace a temporal architecture, a sequence that crosses the human brain from the deep brainstem to the outer cortex over half a century. This animation maps the three distinct phases of the disease. Phase 1 establishes itself in the brainstem between the 3rd and 5th decades of life. Phase 2 moves to the hippocampus in the 6th and 7th decades. Phase 3 reaches the cortex in the 8th decade and beyond. This spatial map is actually a precise sequence of failing cell populations. The disease passes from the noradrenergic neurons of the locus coeruleus to the microglia, and finally to the astrocytes and oligodendrocyte progenitors. They sequentially enter a senescent state. The physical bridge connecting these isolated regions across decades is the SASP. It acts as a diffusible currency, carrying the signal of aging from one failing cell population to the next. Notice that no single senescent cell type explains the entire pathology. The biology changes in every decade. The disease is defined by the sequence itself. Mapping this front follows the disease through a specific chain reaction, with every link joined by these secreted chemical signals. We begin with phase 1, located deep in the brainstem. The very first cells to show pathology are the noradrenergic neurons of the locus coeruleus, starting as early as a person's 20s or 30s. These neurons are exceptionally exposed. They fire constantly to maintain arousal, and they sustain massive, highly branched axonal networks. This relentless metabolic demand keeps the cell permanently near the ceiling of its mitochondrial capacity. This extreme workload generates chronic oxidative DNA damage. To repair those double-strand breaks, the nucleus hyperactivates a repair enzyme called PARP1. PARP1 consumes NAD+ to perform repairs. This chronic hyperactivation creates a catastrophic drain, depleting essential metabolic currency. The neuron cannot divide, so NAD+ collapse forces amitosenescence, marked by mitochondrial dysfunction. It begins broadcasting a low-grade SASP whisper into the parenchyma. The clinical silence of early Alzheimer's is an illusion. That inflammatory whisper is the first appearance of the disease's secreted current, running through the brain decades before a single amyloid plaque forms. That secreted current does not stay in the brainstem. The locus coeruleus projects long axons directly into the forebrain, creating a physical delivery mechanism called the SASP bridge. Through this projection, the resident immune cells of the hippocampus, the microglia, receive a dual hit. As the senescent neuron degrades, it stops supplying the noradrenergic signal that normally keeps microglia calm. At the exact same time, it delivers its inflammatory SASP primer. The microglion loses its brake and hits the accelerator simultaneously. This chronic stress forces the forebrain microglia into phase 2, becoming what is known as a dystrophic bridgehead. Neuroimmunology debated their exact role. One camp claimed they failed clearing. Another argued they attacked healthy synapses. This animation proves they are doing both simultaneously. The cell loses its constructive function, a blocked phagocytic intake, and gains a destructive inflammatory SASP output. Clearance and competence and pro-inflammatory output are two defining faces of a single senescent program. The debate over whether microglia fail or attack dissolves once we define this cellular phenotype. At the exact site of pathology, these two behaviors originate from a single biological program. During the proteolytic turn, the microglial SASP matures. The secretome's profile shifts, taking on a tissue-digesting character. This phase relies on three arms: matrix metalloproteinase enzymes, fenton catalyzing iron from failing glia, and complement proteins tagging for removal. This chemical convergence rapidly strips the perineuronal net, the primary defense structure shielding the cortex's parvalbumin interneurons, the most metabolically extravagant neurons in the forebrain. With the net digested, the cortex enters phase 3, senescent disintegration. The stripped interneuron now faces a barrage from a secondary wave of surrounding senescent cells. Senescent astrocytes enter the fray. They downregulate their critical glutamate transporters, withdrawing the chemical buffering the naked interneuron desperately needs. The result is pure excitotoxicity. At the same moment, local oligodendrocyte progenitor cells are driven into senescence, failing to differentiate and completely halting the remyelination of the damaged region. The terminal phase of Alzheimer's is a fatal convergence. Multiple senescent secretomes fire at once onto a single, defenseless neuron. Thus far, we have tracked a linear, anatomical front moving from the brainstem to the cortex over 50 years. But beneath that sequence runs a single, unifying tissue economy. The heavy SASP burden from phase 2 and phase 3 drives local macrophages to drastically upregulate CD38. This ectoenzyme acts as a massive systemic sink, aggressively destroying NAD+. That depletion feeds back, deepening the exact bioenergetic starvation that ignited phase 1. This loop turns age dependence into a self-amplifying engine. The accumulated senescence of the end feeds the metabolic scarcity of the beginning, closing a one-way biological front into a fatal, accelerating circle. To translate this timeline into medicine, we have to grade the evidence rigorously. We must actively guard against marker credulity. Spotting a generic marker, like lipofuscin, in a brain slice, does not prove a cell is driving a disease. It could simply be a dying cell accumulating waste. This matrix is a validity edger, built to separate theoretical mappings from proven interventions. Phase 2 microglia and phase 3 oligodendrocyte progenitors earn strong grades. In murine models, deploying genetic interventions to selectively clear these senescent cells actively prevented tau pathology and preserved cognition. The causal link is established. The postmitotic neurons of phase 1 receive a moderate grade. The biology is mechanistically tight, but defining a non-dividing cell as truly senescent remains a debate, relying on inference over definitive human trials. Intellectual honesty demands a clear boundary. We treat microglial senescence as a load-bearing, actionable fact, while treating postmitotic neuronal senescence as the frontier of cell biology. This biological map gives us a pharmacology. We now have senolytics, which selectively kill senescent cells, and xenomorphics, which suppress the SASP output without killing the host cell. The application of these drugs relies on a single clinical principle derived directly from the temporal architecture. You cannot kill what you cannot replace. That rule dictates the intervention strategy across the three phases. In phase 1, the senescent cells are the brainstem neurons. You cannot replace them. Therefore, you apply xenomorphics to suppress their toxic whisper and provide NAD+ restoration to support their metabolism. They must be biochemically quieted, never killed. In phase 2, the senescent cells are microglia. These belong to a dividing lineage. You can replace them. Here, you apply senolytics to kill and clear the exhausted immune cells, allowing a fresh progenitor pool to repopulate the hippocampus before the perineuronal net is breached. Phase 3 represents a narrow, complex window. The strategy splits. Senolysis clears out the replaceable oligodendrocyte progenitors, while xenomorphics are used broadly to preserve whatever remains of the matrix. Therapeutic success is tied to the 50-year clock. Senotherapy must be matched to the specific phase, targeting the cell populations that are aging out in that exact moment.